All right. Hi, everybody. This is the session that I call Tool Talk. So in this session, I'm going to introduce some of the lab techniques and supplies that'll be available to you during the lab. And just kind of some reminders to refresh what is going on with the lab. Let's see here. So the question we're going to investigate is, why do the red blood cells appear bigger after being exposed to distilled water? And then we have our two explanations why. So molecules such as protein and polysaccharides are more concentrated inside the cell than outside the cell when the cell is in distilled water. These molecules therefore begin to move out of the cell because of the process of diffusion, but are blocked by the cell membrane. As a result, these molecules push on the cell membrane and make the cell appear bigger. Or the second explanation, water molecules move into the cell because the concentration of water is greater outside the cell than it is inside the cell. As a result, water fills the cell and makes it appear bigger. So one of those explanations is correct and the other is incorrect. So during this investigation, you will have to design an experiment to figure out which one is the correct one and which one is incorrect. So I am not going to tell you how to do it, but I am going to tell you the materials you'll have, and I'm going to show you how to use those materials. So here's our material list. I have starch solution, distilled water, beakers, graduated cylinders, balances, dialysis tubing, safety goggles, and aprons. All right, so to start, I'm going to show you the dialysis tubing. So I just have it soaking here in some water, and I will soak it for you before class. All right, so here is the dialysis tubing. And so it looks like a strip, but if you open it up, it's actually like hollow inside. So I'm just going to kind of play around with the edges here to get this to open. Okay. So here we go. It's starting to open. So this makes an open column. Okay. Now, it is completely open, so if I'm going to put anything in here, I need to tie off the edges. So there are a couple options to seal off the dialysis tubing to make it a bag. One option is you can just tie a knot. So I'm going to twist it, and then I'm going to tie a little knot in the tubing. Okay, and when you tie a knot, you need it to be like a really nice tight knot because you don't want any of the liquid you decide to pour in to fall out. So now I have an opening and it's closed off, so I can pour a solution in here. And it's going to depend how I want to test it, but I can either put distilled water in here, or I can put the starch solution in here. And we'll just pour it in. We can either pour it in from a beaker or a graduated cylinder. And this, like dialysis tubing, it's very small. And so you're going to pour very slowly because it's not going to hold a lot of liquid. Okay, so I'm just going to pour some liquid in here. Okay, so there I have liquid in my dialysis tubing. I might try to make a little bit more. Okay, so I'm going to fill it up as full as I can, and then I'm going to show you a second option to close these. I also have some dialysis tubing clamps. So I'm going to twist it again. And then here is the tubing clamp. I know it's hard to see because it's clear, but all it is this clamp, and it has this opening here. You're just going to squeeze it really tight until it closes. So I'm going to put the twisted part of the dialysis tubing into the clamp. Okay, just like so. And then I'm just going to push it down until I hear the snap. Okay, so now here's my dialysis tubing. I have my liquid inside. On one side I have a knot, the other side I have the clamp. Either option is fine. Okay. The key is I want to set it up so no liquid falls out. And then from there, I can get a larger beaker. And again, the liquid is up to you. I can either go in distilled water or I can go in starch solution. And then it needs to soak. Okay, so in this time I'm going to see, is the water or the starch, is it going to go in? Is it going to go out? What's the direction? And then you also need to ask yourself, how are you going to measure this? Because you're not going to be able to see any transfers of liquid with your naked eye. So how are you going to measure this? How are you going to generate your data? So I hope that this has helped seeing the tool talk. Here's another example of just like another dialysis tubing filled with a liquid. And so now I want you to start brainstorming. What are you going to do? How are you going to test each of those explanations? Okay, so these that we talked about at the very beginning. Okay, how are you going to test those? So think about that. Before you come to class next, I want you to think about what would your procedure be. And I want you to think about how are you going to collect your data and what should that data look like.